Pleased to welcome Professor Deborah Fygenson. She did her undergraduate at MIT, her PhD in physics at Princeton with Professor Albert Liebschauer. And she was apparently his first student to pursue a biophysics-related project in microtubule dynamics. And then she did a postdoc in the laboratory of Professor Goodman at University of Southern California, where she got a fellowship from the Jane Coffin Child's Memorial Fund. And then in 1998, she moved to UC Santa Barbara and started as a professor, and has received prestigious awards such as the Sloan Fellowship and his career. And without further ado,
thank you, Walter.
Deborah.
I am so happy to be here.
It's our first in-person since it's non-local in-person since 2020.
Yeah. And first time I'm non-local since then too. And I hope that it's a sign that it's all coming to an end. Because I've just had a great time visiting and meeting people and actually seeing labs of other people in person. It's so much better. And it's a real pleasure to be able to tell you about research that's going on in my group. Today I'm going to be telling you about the work of one of my graduate students, Jinwei Kai. We call her Amber, so I might slip into calling her that. But to give you the context that sort of covers all the research I do, it really comes down to machines, basically. I like machines. We find machines all around us, and they're very purposely built things, right? They're made with intention, and they harness what we understand about the physical world. And if you need a machine that you don't quite know how it works, it's okay, because somebody knows how it works. Somebody invented it, somebody produced it, somebody's probably improving on it as you speak. What really fascinates me, though, is that it's not just our everyday lives that are full of machines, but it's our very selves. Like, our bodies are full of machines. And I don't know if you know this book. It's one of the first ones I was told to read when I was told that biology is the future and we have to start studying it as physicists. David Goodsell puts it very well, that the machinery of life is one remarkable common denominator. That we're not just, as humans, similar to primates and lab rats, but also to trees and slime molds. Because once you get down to the sub-cellular level, machinery is remarkably similar. So I think that the machinery of life offers a grand challenge to physics. So, sure, you can ask what machines exist, and you can ask how do they work, but those are actually challenges to biology. When it comes to physics, we want to know why they're built the way they are. What are the principles of their design? And it's my contention that to understand these, I'm sure I got it from others, but to understand them, eventually create molecular machines that serve us, not just harvest them from biology or tweak them from biology. I say we have to reinvent the wheel, right? And the axle, and the lever, and the spring. And we just have to do this work of trying to build with purposeful intent based on how well we understand the physical constraints. So what I'm going to tell you about today is a baby step in that direction. I think it's a well-posed one, and we can motivate it in a lot of ways. The tool is really not much a machine as a device, but the tool is going to measure DNA bending. And you can ask why. And the reason is accessible. We can do it. And it's been of interest for a long time. There are a lot of tools that do it. I'll tell you first that it was discovered back in the 1980s. So people have been studying this for 30, 40 years now. And it was discovered by looking, I don't know if any of you know what a gel is, but basically you run molecules through a gel, a network, and they separate according to their mobility, obviously, but according to their size or charge. And if a protein is bound to a molecule, it makes that molecule run more slowly. And what people noticed back in the early 80s was a very unusual thing about the DNA of this bug here. I've even forgotten its name. But there's DNA in the nucleus of the bug, and then there's this other thing called the kinetochore, another little pocket of DNA at the other end of the bug. And what happened, these guys were running gels of different pore sizes, different percentages of the polymer that makes up the gel. And the shift in the DNA between this group of DNA and that group of DNA was different depending on the pore size. So that's kind of unusual, right? Because you'd think that the pore size would cause a uniform kind of change to everybody's resistance to motion or mobility. But you can see that the DNA from the kinetochore was very close to that of the nucleus at the large pore size, and then got a lot slower, and even slower still as the pore size got small. And so people glommed onto this, and they after I think about 20 years of work, figured out that it's because some pieces of DNA in their sequence are bent. So there's an intrinsic bend, and in this case it was due to a string of the letter A. You know, DNA has A, C, T, G. This was a set of 6 A's, then 4 C's or T's, and another 6 A's, and then it went on like that. That pattern in the genome caused bends that gave the DNA this unusual mobility feature. And people have learned since then that those kinds of bends are crucial to packing up the DNA so that it can be replicated and separated, and to packing it or unpacking it for transcription, and to attracting the molecules that glom onto it to initiate the process of making protein based on its recipe. So there's been a lot of effort to understand DNA bending. It seemed like a reasonable thing to target. And there's a fair amount of existing approaches, not just the gel shift assays that we just told you about, but you can do X-ray crystallography, you can do cryo-TEM, you can do AFM, and a few other things too. But there's various limitations in these, like, low angular resolution. You don't really get to see from a gel. You can see it's retarded. You can maybe hope to calibrate on some known bends, but you really don't know what kind of bend you're getting. You can go to really high resolution with X-ray crystallography and even cryo-TEM, but now you've got a lot of complexity and cost. So the chances of you just exploring a lot of sequences to figure out what is going on in the genome. See, the thing is, if we could read, this is like a whole nother meta-information to the genome. You read the sequence out, you know the recipe for a protein. You read the structure out, and you can understand which protein gets made when, because these regions are important to bringing together parts and getting that complex that translates the DNA initiated. So it's great that you can measure it, but if it's so difficult, you're only going to measure it, one PhD at a time, and it's going to take forever to really decode it. When you go to AFM, you might have that perturbing context of the surface. You got to hold it down, and you can work ways, but then you have to make some assumptions. It's a little bit less nice than, say, cryo-TEM, where you freeze it in the water and you see the natural shape. And all of these things are non-dynamic. So if the dynamics makes a difference, say the stiffness of a particular bend is important, fluctuations in that bend are crucial, then you don't get it from here. Now there's one other technique, which is single molecule fret. It lets you watch in solution. Well, you kind of have to tether things down on the surface, but you can do it pretty gently. And it gives you time resolution, which none of the others did. But you have to actually look at fairly big bends, and those aren't necessarily all that common. And it's also kind of complicated. You don't do it with the microscope you buy off the shelf. So it seems like this was an angle with what I know how to do, which is build things out of DNA. I could start to build a tool with my own purpose in mind, and make some of the insights into what does it take to engineer meaningfully at this level. So that's what we're going to do. And the tool that we're building is called a nunchuck, for reasons that I kind of hope are obvious, right? It contains a tiny piece of double-stranded DNA here at the center. So that's the typical double helix that your genome is made out of. And that links together two pieces of DNA origami that are seeds in this case for what comes next. So DNA origami is a technique of weaving with DNA. You basically make a cloth out of this molecule because you can get very long strands of DNA from a creature's genome, and then you can buy short strands from about a dozen different companies now for pennies on the base, and you mix them together and they'll fold up in a way that you design, because the sequence of the short strands can be designed with specificity for parts of the long strand. So this is an Ox DNA viewer mock-up of what our seed looks like. It has little bumps because we wove a cloth that's a rectangle and curled it up and sewed it together. So in order to make sure, we didn't do that by hand, it's sort of self-assembled, so we had to bias it to curve in one direction. So we put some little DNA bumps on one side of the cloth to make sure that that's the side that would be on the outside. And with this technique, we can now say which side is which and attach things to it. And this was the work of a grad student at Hopkins in the lab of a collaborator of mine named Rebecca Schulman. And Rebecca invented the seed, and Abdul, he put it together into the nunchuck with us. He characterized the seed's nucleation for his PhD. And so we used Abdul's protocol, and we can grow these seeds, which are only 75 nanometers or so long. We can grow them out the building. We can grow them to microns, many microns in length. And that's really powerful because now we're optical. Now we can watch in solution in real time. So the diameter of this tube is the 10 nanometers-ish scale, and the length is on the 10 micron scale. So we've got a good factor of many hundreds in aspect ratio. And we get to look at these seeds under the microscope using fluorescence. Often the challenge there is to make something bright enough. But because these tubes are grown from small clusters of short oligos that are relatively cheap, we can get one of those oligos to have a covalent dye on it. And now we have a high density of dye molecules all along our tube, making it bright as hell. So it's easy to see. You do this research with your garden variety microscope. We use a little clever technique of labeling one side of it with 2 dye molecules of the same color per tile, and the other side has 2 dye molecules of different colors on their tiles, one of which is identical. So in one channel under your microscope, you see a bright arm and a dim arm, and in the other channel, you see only the dim arm, and this one's gone. That means you have a very easy way to confirm that you've built what you intended to build, because that type of a structure won't occur spontaneously. Tubes will form spontaneously even without seeds, but they won't be 2 colors with a junction in between. So that's our structure, and this is what it looks like. And this is a movie. So we watch it under the light microscope. You see the bright arm and the dim arm. And it actually diffuses around a lot more than this. We're just tracking it, or we actually just piece it together to be centered afterwards, really. But you can see that it is maybe more flexible at the interface between the bright and the dim arm. Maybe you see that. And these arms, they're pretty stiff, right? They're about 4 microns long in this case. We've measured the persistence length of DNA nanotubes of all types, and this one is persistent, so totally decorrelates its orientation of the ends of its arm on a scale of like 20 microns. So at 2 microns, it's really pretty straight. I wouldn't mind if it was 4 times straighter, make my angular resolution better, but it's pretty good. So we're going to measure the bend angle. So this is how we define the bend angle, with a theta that's 0 when the object is straight, and 180 when it's completely folded over. Okay, now we have to get to the point of measuring that bend angle. All we have are pictures, and every time you have to get data out of pictures, you're in for a bit of work. So one way to do it is to do filament tracing. You basically take every image and you track along the bright gradient until you see the length of it. And then once you have a skeletonized version of your filament, you can identify a transition from bright to dark along that skeleton. That's your junction. And then we would take a series of tangent vectors along each side, compare that on both sides, and decide. We only look over about a micron, so it should be stiff enough to give us a unique direction. And we take the dot product and normalize it, get out the angle. It's a laborious procedure. And when Amber started, it was like dozens of days between taking some data and having a data set. And she was very focused, and she worked really, really hard, and she freaking gave herself like a work injury clicking the mouse around, and was out of the lab for like 6 months because she just needed to recover. It was terrible. So I'm like, no, we can't do this anymore. Time to do some machine learning. And machine learning is perfect for this, it turns out. I'm pretty much a neophyte in it, but we've made it work for us. So now it's like take data in the morning, see data in the afternoon, which is amazing. Our machine learning is, with these machine learning algorithms, you have to train them. So you need lots of images to train them from. And, well, you need lots of images you know the angle of. So if you need 100,000 images that you've taken the angle this way, you're still up the creek. Instead, it was an undergraduate in the lab, Sebastian, who came up with an algorithm for making fake images. Our images are so simple, we can make fakes easily. And we've gotten very good at this, at making the fake images have all the problems of the real images, with the occasional other structure coming by and getting things distracted with changes in the relative intensity of the background and different kinds of blurriness. So we can make hundreds of thousands of fake images in the blink of an eye. And because we make them, we know what angle they have. So now we can train up a neural net really well. And oh wow, our neural net got a lot better with COVID, because when you can't be in the lab, you can still be on the computer. So we have, the neural net results I'm showing you here are not quite as professional as those, but this worked really well. We get out a bend angle for every frame. We can process thousands of frames, and we even get a confidence score. And we could do some regression on that, but we don't bother. Right now, what we did to first convince ourselves that we were doing something we trusted was we tested what the neural network said with what the more manual filament tracing sort of algorithm said. And they agreed pretty well. In fact, I was a little disappointed that it kind of looks like that center blob is kind of off axis. Turns out that the filament tracing program is a bit biased towards things that are more smooth. And we've been able to do, so we sat some undergrads down and they did filament tracing on fake images. And we figured out that we can really trust the neural net a little bit better than the filament tracer. But we've got very reliable extraction. So we can take data at, well, you usually take it about 1 hertz, but we can go up to 10 hertz and collect thousands of frames and analyze them quickly. So this is what the data look like. What you see here are the angle histograms from movies of 17 different nunchucks, one histogram per nunchuck. The number of frames is written in there, so you see these were 500 frames each. That's a pretty good number of measurements, and they all had the same linker. They're nominally the same molecule. And indeed, a lot of the histograms look alike, but out of the 17, 4 of them really look weird, right? Some are weird that their average position is non-zero, clearly non-zero. Others are weird in that they have just a very huge dispersion. That'll come back later. We ended up sort of plotting all of them on a mu sigma plot and noticing, using clustering algorithm to say, okay, these guys are something and these guys are outliers. And okay, we're going to ignore the outliers for now. One of the complications of this type of measurement is that we can't tell the sign of our angle. And that's because, okay, I forgot to mention that we have to confine these things to 2D so that they stay in the focal plane while we watch them for many minutes at a time. But that confinement is to keep the tubes, which are microns long, kind of in the plane. They're microns long, but they're 10 nanometers thick, so there's really not much preventing them from rotating at all. And they surely thermally rotate about their long axis as much as they flop around. So we can't really keep track of what direction any one nunchuck is bending in. So what we're looking at is a normal distribution, a Gaussian distribution that is folded at 0. Okay, so that's fine. We fit to that Gaussian distribution with the folding back at 180 as well, but we don't really have much data out at 180, so that's not such a problem. And we can find mu and sigma by doing those functional fits. And what do? With this lets us measure the stiffness of a linker. Because it's a fun fact, not really well known, but easy to actually do the math once somebody shows you, that the average of the cosine of an angle that's normally distributed, about 0, goes as e to the minus the width of its distribution squared over 2. And we all know that for worm-like chains, I don't know if we all know, I mean, in our field where we study polymers all the time, worm-like chains have what's called a persistence length that is essentially defined by how decorrelated that angle gets. And so it goes as, well, exponentially decays away with length, and that characteristic length is the persistence length. So those look a lot alike. So we can actually say that if we know the variance in angle of our junction, we can know what the persistence length of the linker at the junction is, right? Because we're looking at the tubes that come off the ends of this piece of DNA are essentially telling us the tangent at the end of the linker. The linker can be doing all sorts of things in between. We're really only seeing the direction of the part that's attached to the tube. So we decided we could check and see if our machine is reliably measuring the angle that we want to measure by making sure that this relation holds. So we're going to plot variance in radians squared versus linker length for the different linker lengths that we explored. And there you see the data, and you're like, well, it's supposed to be a line. It looks maybe not quite like a line, but it's close. But once we realized that we have an issue of steric hindrance, we could focus in on the tubes, on the machine, the tool that was built properly. So because double-stranded DNA is helical, there is a phasing. Because the linker goes into a point on the rim of our cylinder, if the two arms are separated by an integral number of helical turns, they'll both be on the same side of the linker. So we have to separate our arms by a half integral number, odd half integral number of helical turns. They have to be out of phase. When you flex around in this way, it takes quite a bit of an angle before the two tubes will even see each other. In this way, the two tubes can see each other pretty quickly because, remember, they're 10 nanometers long and that piece of DNA in between them is on the same order. So you get some steric hindrance. So accordingly, the seeds that were made in phase, well, if they were short enough to be stiff enough that it doesn't flex much, it was fine. They got sort of maybe the kind of bend angle distribution you would expect. But as they got longer, maybe it was the steric hindrance that kept them from seeing that angular distribution grow the way you would expect. So if we focus just on the out of phase seeds, we can find how it depends on linker length, and we end up seeing that slope is 130. Our confidence with just 4 points over just this distance is 20%, but we're getting a reasonable number. We really are seeing the effect of the linker, not other weaknesses in the seeds or the tubes or anything like that. So I would say we've been successful at magnifying up this nanometer scale conformations out to the micron scale to read out at least this linker. This is an interesting feature, lambda 0. So I'm calling linker length lambda, and you would expect that at 0 linker, if you have something stiff, you would not have much of dispersion and angle at all. We've actually made solid tubes that are half two-color tubes, so we know that actually just the fluctuation of the tube itself gives you something like 0.1 radians squared as variance. But here we're getting quite a bit more than that, and the intersection, the x-intercept, place where there really is perfect stiffness, could be interpreted as perhaps our linker, the way that we connect it into our tubes, has some play too, so that the overall double-stranded single double helix nature of it penetrates about half a helical turn on each side. It's possible. But looks like it's working. Let's see if we can use it to measure an angle that's not 0. So for this, we go back to what everybody who measures angles test themselves against, which are these poly A tracts that I talked to you about before. And here, these are how the in-phase A tracts would cause a bending, as determined by a variety of other techniques. So we also looked at now the mean bend angle from our distributions as a function of the number of A tracts that were placed in phase inside our linker. And indeed, we saw a change in mean bend angle proportional to the number of A tracts. That slope was about 17 degrees, which is what folks measure, plus or minus 1 degree, which is just as good as what people get out with the fancier techniques. So it works also to give us the intrinsic bend angle. And of course, if we're going to look at many A tracts, we have to make the linker longer, or we can make 6 A tracts is going to require a long linker, about 60 bases, where you could make something with 3 A tracts half the size, and we actually did this at many different linker lengths, and they all plop out there. We could only fit, we did this with 1 linker length. So we controlled for linker length as well. And that meant that we also had the ability to compare them on this plot. We could look and compare linkers with the same density, the highest possible density of A tracts as a function of linker length. So we were using some which had only 2 and 3 and 4 and 5, and looking to see how their mean bend angle was changing. And these are those data points. So, yeah, this is pretty good. We had to take the same intercept, but then we could see that all of these had much less angular variance than the ones that didn't have 8 tracks. So it's not unexpected. People had done simulations and some calculations indicating that 8 tracks were probably stiffer than regular DNA, but I don't think anybody had measured it before. So we've got our tool to the point of actually making a measurement that hasn't been made before. And it's kind of interesting. They're 50 to 100% stiffer. I mean, they're significantly stiffer. So that was kind of nice. I don't know why I have 2 of the same slide. Oh yeah, so here we see those 2 together. So that's encouraging. And now we have to take a critical look at this machine we're able to build and ask, what are its limitations, what are its prospects, and what have we learned? So one of the key limitations is linker length. We can't look at linkers that are too short because then, the tubes bounce into each other at too small an angle. And there's a detail in the way we design these where we rely on DNA hybridization to make the linker assemble. And if you get a lot shorter than 25 bases, it doesn't hybridize easily. We could engineer around that, changing the way that we make them. So it's not a hard limit. It could be pushed lower, but not that much lower, because at some point those tubes are still a challenge. And those tubes come up again at the high end. If you put something that's way too floppy, well, you're not going to see anything meaningful because you'll have exceeded the correlation length, the stiffness of the double-stranded DNA. And also, as they get longer, they might actually fluctuate around enough that the tubes also start hitting each other. So there's something there, but it's not fatal. 60 bases, 50 bases is enough to put a lot of interesting things inside. And there's another challenge that comes from the fact that our angle distribution is folded. This is what the folding looked like. So here's 0. If I have a finite mean bend angle, that means that all of the measurements here appear as positive angles as well. And so those have to be added together. And then anything that goes beyond 180, our things are stiff enough that they don't go beyond 180, but it could have the same sort of problem at 180. Consequence, even for the kind of angular distributions we get with sigmas of the order of 30 to 50, is that you can have 2 very similar looking distributions, one with mu 0 and sigma like 50, and the other with mu 20 and sigma 40. So that's just the math of the Gaussian distribution that you fold over at 0. It's got this interesting degeneracy to it. And so we have to work some way out to distinguish these. I would say our prospect there is maybe to get away from the perfectly straight arm and try to build in a kink. If our arms are actually more kinked, then it's not as easy for it to rotate about its long axis. And now we might be able to tell the difference between a positive angle and a negative angle and get better resolution in those small angles. And the main prospect, I would say, of this technique, though, is that it is real time in water, right, where proteins that are going to interact with DNA can live and work happily. So maybe we can see dynamics. And this is an interesting question because don't those arms slow things down? And I'm sure they kind of do, but energetic-wise, they don't cost us anything. They diffuse into the lowest energy conformation. But dynamically, we're sort of wondering what's going on. You might have expected, given the rotational diffusion coefficient about the short axis for a cylindrical object, that the angular diffusion coefficient would scale actually with arm length cubed. And we're only seeing a linear kind of scaling. So we don't know quite what's going on here, but we do sometimes see changes in angle that happen quite quickly, like 100 degrees in less than a second. So the hope is that we can open up this world of understanding protein-induced bending, because that's where all the code, the meta information of the genome comes in. The regulatory proteins bind and bend the DNA to initiate transcription. If we have enough opportunity to see rapid rearrangements. So that's one of the futures. So our next targets, things that fit inside real well in that 27 to 58 base length, are defects that are commonly found in DNA and would be interesting to know what kind of angle they make. We have some data from the other techniques that can tell us the angle, but also how stiff that angle is. Like when that angle gets made, how much variance is there in it would be really interesting, and what its lifetime of those bent states are. So we're looking at these two defects now and finding some sort of surprising results, that they're not as bent as you might expect. It's easier for DNA to be straight than it is for it to be bent, it's starting to look like. And we're also starting to look at regulatory proteins, because we can fit their binding sites inside of 57 bases. We're particularly focused on integration host factor because it has a total U-turn effect. So we should be able to really see the DNA bend around. So what does this tell us about machinery at the macromolecular level and the building of it? I thought I'd put a slide in here just a second ago that I wanted to share with you all. Let me just see if it's here. I didn't. So I'm going to grab it over for just a second, because there is a big lesson to be learned from this. Give me a moment. It's basically all that clustering and those outliers that I wanted to share with you. This is kind of a complicated graphic, but it gets the point across. This is the mu sigma map for the various numbers of A tracts. And these are the 9, 10 different nunchucks that we looked at to get our measurement for that one 58 base pair A tract, 6 A tract linker. And what you'll notice, and if you'll notice it for any one group of data points, is that we can never get away from seeing individual nunchucks that are totally different from each other. And we have tried. We have tried to get rid of all excess DNA that could be causing some transient binding. We put in proteases to eat up any protein that might be around, that might be causing binding. I mean, at the single molecule level, we have so much sensitivity and so little control, it's mind-boggling. We've tried digesting away defects of DNA with the right enzyme that finds little loops and bulges and eats them up, and should, essentially eliminate problematic nunchucks. And it doesn't change the frequency of our outliers at all. We have 20 to 25% weirdos. Whenever you cluster, there's always a quarter of them lying outside. And I think this is our biggest challenge as we try to build at this mesoscopic scale. Because we're used to thinking about molecules, well, atoms we know are identical. They're so interchangeably identical, we have to take that into account when we calculate the entropy of something that's going on. And molecules are also pretty darn identical. They have 10 atoms, 20 atoms, whatever. And if they're not perfectly identical, they give different enough properties, you can filter till you get a very pure solution of a certain molecule. But when you're starting to build out of billions of atoms and thousands of bases, now your structures have a rugged energy landscape that is very complicated. It's what we think may happen is that there are long-lived metastable states that you can get into, and on the time scale of your experiment, even though you think you've gotten to 10 minutes, 20 minutes, that's for something that deals in millisecond fluctuations and effects, you think you'd be ergodic by then, and you're not. That's my best hypothesis. And we don't have, it's a long-lived state. Maybe it comes out again. Maybe it's irreproducible. Can we know what the nature of all those defects are? Can we filter on them in any way? I don't know. I guess what I'm saying is we want to build machinery like biology that does things in this wet and raucous world. We may need to accept one of the things biology does, which is constantly chew up and toss out its machines. They are only good if they work, so the ones that immediately don't work get segregated out, and then they go bad, so you segregate those out too. So I don't know, but I'm wondering if macromolecular machinery is intrinsically a transient thing, if we're going to start thinking about it like the kind of technology we have every day. So that is what I wanted to say, and much of this work is due to Amber, but Abdul and Rebecca helped in the initial design, and providing those seeds was crucial to enabling the nunchuck. Lourdes and Deborah did their undergraduate theses looking at various controls that enabled us to get things reproducible, and Daniel built the first nunchucks in our lab. So with that, I would love to enter into a dialogue and have a conversation. Thank you. Yes, please.
This may seem like a silly question, but what is causing the thing to wiggle?
Here, I should bring
Let me see if I can repeat, or okay, let's have you read so the people on Zoom can hear.
What is causing those machines to wiggle around like that? What's the energy source?
That's temperature.
Okay.
Yeah, that's Brownian motion. It turns out that 1 kT is plenty enough to make things move by microns at this scale.
So,
Just hold it. Yeah, that's good.
like this?
Too loud if you hold it in your mouth.
Oh, I see.
If you're going to hold it yourself, you got to
Okay. I have so it's kind of related. My question. So you were talking about dynamics, and so we expect normally that you have a kind of Fokker-Planck equation, and we know that the diffusion constant is going to be proportional to kT, and we with the dynamics presumably you can see if it's well described by a Fokker-Planck equation with a given potential. That should be your stationary distribution. So, but then you mentioned that you have like sudden turns, which does not seem to be completely compatible with a regular Fokker-Planck equation for the angle. So I'm wondering what is happening here. like you should be able to infer, I don't know, some kind of, just from the dynamics, some kind of potential, like the rates of transition from one state to the other. And so,
Well, maybe it's because the tubes themselves are not fast to respond all the time.
Okay.
So you have to you'll get the fast response more stochastically when the tube itself diffuses in quite the right direction that the fluctuation of the linker is compatible with, and then it will lock. So you'll know that you had that fluctuation.
I see.
But many of them are masked because the linker is able to do quite a bit of rearranging with the tube not.
So but that suggests there is a kind of hidden variable, and then when you what you see is that you marginalize over this hidden variable, and then you get the global dynamics. So, but there should be a way to just look at the data and infer. I don't know, make some
maybe
kind of
there
hidden
is.
Markov model to just infer exactly how many states you are, or something like that.
I would love to look at the data with someone who knows how to do that.
Okay.
That would be really interesting.
That's really interesting.
Yeah, we have lots of data, so that's usually
great.
a good
That
sign.
sounds very interesting to do.
Thank you.
So you mentioned that these machines are confined in 2 dimensions, right? So the question would be, is that relevant for the dynamics? And then if it is not, which I imagine, could you fabricate some sort of arms that in which you can distinguish the orientation as well? I imagine like a delta and some sort of thing like that.
That's a great question. So yes, I think I didn't emphasize it enough. So the space that they're confined to is about 500 nanometers, maybe a little bit more. They're 10 nanometer thick arms. So they go out to microns, and it's more their length that focuses them than anything else. So the structure of the linker is actually a 3-dimensional structure that is free to rotate into the plane and can freely just position itself so that we can see its most bent angle. So yes, I think that we're dealing with the 3-dimensional fluctuations of linker conformation. And I would love to make arms that are a bit more planar rather than just linear, so that the confinement rotationally is limited. It's a bit of a challenge. The structures that we can make out of DNA with a lot of arbitrary features, or they tend to be a bit small compared to 500 nanometers. So we need to make something quite large. The best thing might be a kink in the tube itself. So we have one long tube and then another one growing off. We haven't tried yet, but the statistics of getting 2 things to come together to link up, we have to do that with the short seeds and then grow the tubes. Because if they're already long, it's too hard of a search problem. They don't really self-assemble anymore. And so growing something with 2 different seeds, one to attach to the linker and another one at the other end to make another tube grow off in the other direction, we're coming too far away from self-assembly. We don't get to do it in the large quantities that make this useful. So I haven't maybe had the right good idea. Like you were saying something like a triangle or something, and I don't quite know how I would do that, but maybe. Just growing from one seed is easy. Growing, the 2 seeds capped will make our yield much lower and be harder to find. But we could ideally do it.
I have a question.
Yes.
Have you looked at salt dependence? There's this interest in the salt dependence of the persistence length that's kind of controversial, particularly at low salt. And the measurements from tweezers are all kind of scattered and not that great.
Oh, really?
Have you ever tried looking at salt variation?
We haven't varied the salt. So the origami and the tiles, they pack the DNA in quite close. And it's important to have counter ions that can neutralize that charge. So we have to have some amount of magnesium around. We tend to have about 10 millimolar. But we can play, because biology gives us enzymes that work on the DNA, and we can make these things fully covalent. Well, to as good as the enzyme will work in its yield, but we have been able to ligate up these nunchucks so that they're robust to, say, 60, 70 degrees temperature. So they're pretty well held together. Under those conditions, I think we could exchange the salt and start to vary. But if you need to get to salts, monovalent salts that are less than, 50 millimolar, I don't think it's going to happen. So is the salt dependence that's of controversial lower
it's
than
low.
that?
It's like sub 10 millimolar.
Yeah. I don't know if our structures can be stabilized down to that level. But I'll look into it. It's good. I'm sorry I didn't mute my phone. It's making lots of noises somewhere.
Okay.
Put more?
Okay. All right.
Another question.
sorry.
I have another kind of maybe stupid theory question or from a theory. So is there a way to maybe do some kind of single molecule like things with optical tweezers and kind of measure, like fixing them and just measuring with like trying to move the molecule around with the nunchuck, or would that be too sensitive, you will break it?
Oh, no, I mean, and actually it connects over to your question, and it's why I'm here to see Walter, is because if we can put these in these interesting confining cavities of his, maybe in some that have a geometry that has an angle to it, maybe we can play that.
Okay.
I don't think that an optical tweezer will have enough dielectric constant to grab.
Okay.
We'd have to put some kind of a biggish bead or something to really hold. But I think in the kind of systems that Walter works in, we could get these things confined. Yeah, that's good thinking.
Thanks, speaker again.
Thank you.